Cellular respiration is the catabolic process in which organic molecules are broken down to create usable energy via an electron transport chain. This process requires oxygen in humans and most other organisms and produces carbon dioxide, water, heat, and usable energy in the form of ATP. While many different organic molecules, sugars, amino acids, and lipids, can be used in cellular respiration, glucose is used as the prototype. Thus, the equation for cellular respiration is C6H12O6 plus 6O2 leads to 6CO2 plus 6H2O plus energy, the reverse of photosynthesis. This reaction actually occurs in multiple steps. Glycolysis is in the cytoplasm, pyruvate oxidation and the citric acid cycle occur in the mitochondria, and oxidative phosphorylation takes place over the inner mitochondrial membrane. Together, these processes power cellular activities, from flagellar movement, muscle contraction, through the breakdown of organic molecules to produce ATP.
Glycolysis is the metabolic process of breaking down sugars to generate energy. Glucose first enters the cell's cytoplasm and is transformed across two main phases. The first part requires energy, in the form of ATP, to trap the glucose molecule inside and modify it through a series of steps. The second phase extracts energy from electrons and harnesses it as high-energy NADH and more ATP molecules. After multiple enzymatic steps, 2 3-carbon sugars, pyruvate, are produced and ready for transport to the mitochondria for entry into the citric acid cycle. Glycolysis begins with a single glucose molecule. Using ATP, the enzyme hexokinase transfers a phosphate group to the 6-carbon sugar to produce glucose 6-phosphate, which becomes trapped inside the cell due to its negative charge. Next, the enzyme phosphoglucose isomerase catalyzes the conversion of the phosphoglucose into one of its isomers, fructose 6-phosphate. The phosphofructose can now be phosphorylated by a rate-limiting enzyme phosphofructokinase to produce fructose 1,6-bisphosphate. Finally, with 2 phosphate groups attached, the sugar molecule is cleaved by aldolase into 2 3-carbon isomers, glyceraldehyde 3-phosphate, or G3P, and dihydroxyacetone phosphate, DHAP. Another catalyzing enzyme, triose phosphate isomerase, converts the DHAP into G3P to yield 2 molecules. Thus, during this energy investment phase, a net total of 2 ATPs are used to split one initial glucose molecule into 2 smaller sugars.
In the second half of glycolysis, the 2 molecules of glyceraldehyde 3-phosphate, G3P, are oxidized with the reaction catalyzed by the enzyme glyceraldehyde phosphate dehydrogenase, and a phosphate group is attached to the unstable sugar, forming 1,3-bisphosphoglycerate. As a result, 2 high-energy electrons and 2 protons are released and picked up by the carrier NAD+, forming 2 NADHs and hydrogen ions. Phosphoglycerate kinase then transfers a phosphate group from each 1,3-bisphosphoglycerate to ADP, creating 2 molecules of ATP and 3-phosphoglycerate. Next, the enzyme phosphoglycerate mutase converts this molecule into its isomer 2-phosphoglycerate, which allows the enzyme enolase to release a water molecule and form a new double-bonded structure, phosphoenolpyruvate, or PEP. With the help of pyruvate kinase, the phosphate groups are removed from PEP and given to ADP, generating 2 more molecules of ATP along with the end product, pyruvate. Thus, at the end of glycolysis, a net total of 2 ATPs are produced, along with 2 NADHs and 2 pyruvate molecules. With oxygen present, pyruvate can be broken down further, while NADH can pass its electrons into the electron transport chain to regenerate NAD+.
Through the anaerobic process of glycolysis, which occurs in the cell's cytoplasm, 2 ATPs are used to divide one glucose molecule into 2 pyruvate and 2 water molecules, and create 4 ATPs, 2 NADHs, and 2 hydrogen ions. So, in the net reaction, where compounds that are both produced and consumed are removed, the reactants are 1 glucose molecule, 2 ADPs, 2 inorganic phosphate groups used to make ATP, and 2 molecules of NAD+, and the products are 2 pyruvate and 2 water molecules, 2 ATPs, 2 NADHs, and 2 hydrogen ions. Sometimes this equation is simplified to show only the major players of glycolysis: glucose, 2 pyruvates, 2 ATPs, and 2 NADHs.
After glycolysis in eukaryotes, the charged pyruvate molecules enter the mitochondria via active transport and undergo 3 enzymatic reactions. The process is catalyzed by a complex, the pyruvate dehydrogenase complex, composed of 3 enzymes. In the first step, the carboxyl group is removed from pyruvate and released as carbon dioxide. The stripped molecule is then oxidized to an acetyl group, and the released electrons are then picked up by NAD+ to form NADH. Finally, the acetyl group is transferred to coenzyme A, where it attaches to the sulfur atom to create acetyl coenzyme A, or acetyl-CoA, which moves into the citric acid cycle to be further oxidized.